Hello, everyone. I'm Laura Splan, and I'm an interdisciplinary artist exploring the interconnectedness of cultural and biological systems. While sheltering in place last year, I collaborated with scientists to create molecular animations of the SARS-CoV-2 coronavirus. I often repurpose tools and techniques of science to engage with them both creatively and critically while searching for new possibilities. And I choreograph poetic confrontations with science inside the gallery to foster deeper engagement with science outside the gallery, while framing complex biomedical issues with provocations of curiosity and wonder. The collaboration culminated in an exhibition of the animations at the Brooklyn Army Terminal that was accompanied by an immersive soundscape inspired by the genetic sequence of the coronavirus. I invited scientists and lab technicians to play the SARS-CoV-2 nucleotide sequence on guitar in recorded Zoom meetings. The recordings were made by simply prompting participants to play A on guitar 33 times, the number of adenine nucleotides at the end of the mRNA sequence. I often leverage the material artifacts of biotechnology to reveal poetic subjectivities of the seemingly mundane. I attempt to destabilize and reframe our relationship with the everyday, and even redefine what the everyday means. I created this lace SARS doily in response to the first 2003 SARS outbreak. This project was grounded in the intricacy of textiles and its associations with the familiarity and comforts of domesticity. But in the speculative narrative of the work, collective traumas born from disease, from epidemic, from pandemic are rematerialized in an heirloom artifact to be passed on from one generation to the next. I often try to compel an intimate engagement with detail, calling into question how things are made and what they are made of. And I use processes and tools that challenge notions of what is made by hand and what is made by machine, what is natural and what is constructed, and what is science and what is culture. This project led me deeper into the layers of scientific representation and the ways that biological forms are translated and abstracted with technology. In 2018, I was invited to be an artist in residence at the Science Center in Philadelphia. During my residency, I had the opportunity to shadow scientists, as well as learn software that they were using for antibody research. With a sensibility informed by textiles, I began to unravel protein structures using a feature in the software called sculpting. In response, the software rendered uncanny disturbances in the form of sometimes spastic and sometimes sublime movements, which I captured as a series of animations. I showed this sculpting feature to scientists at the lab who began using it in their own research, allowing them to visualize structures in new ways. During the residency, I was also struck by the lab's use of non-human species for the production of antibodies. My conversations with the scientists led to receiving over 200 pounds of wool from laboratory llamas who produce antibodies for human drugs, including vaccines. Since then, I've been spinning their wool into yarn. And I situate sculptures made with the yarn among other artworks that examine the hidden systems and invisible labor of biotechnology. This work questions notions of the presence and absence of bodies, evoking the mutability of categories that delineate their status. While processing the wool, I would often find clumps of feces, which I began to collect for this networked laboratory mixer. The device agitates when Twitter hashtags associated with the culturally contested status of science are tweeted. When taking office, the Trump administration advised how to improve the chances of receiving research funding with the suggestion to avoid phrases like diversity, transgender, and science-based. Here, the mere mention of hashtag vaccination agitates the tubes filled with laboratory llama feces. Behind another sculpture in the gallery, faint text on a wall invited viewers to come close to read, "Our distance allows our intimacy." The sculpture blows a breeze in the viewer's face as they read the text. The speed of the networked fan intermittently adjusts based on the wind conditions near a farm in rural Pennsylvania. The farm is actually the 600-acre biological laboratory that gave me the wool from the shearing of the llamas. I'm now experimenting with dyeing this yarn using genetically manipulated E. coli to produce pigment for a new project exploring interspecies entanglements. I'm looking for collaborations with biological laboratories to help me continue these bacterial dye experiments and partnerships with vivariums to source artifacts from animal-derived biological products. I'm also seeking opportunities for exhibitions and public programming that engages audiences in the sublime beauty and contested territories of the biological world. You can click the links in the chat to get in touch with me, and I look forward to connecting with you.
It's amazing. Thanks so much, Laura.
Oh, thank you.
Do any of my fellow Paulas want to jump in?
I have to keep unmuting. I'm just going to jump in. Is that cool, John? So there's a lot that's very interesting there. On one hand, you have this very technological side, the software, the science, but then extracting from the science, this sort of unglamorous throwaway, right? The poop and the wool. And so I wonder how you're navigating these two different threads. Are you more interested in the sort of unglamorous garbage? Are you more interested in the probing the science and the technology? Or for you, is there not such a clean separation between those?
Yeah, there's not really a clean separation. I'm often kind of foraging for materialities within both. And in order to kind of enrich the understanding of the material, I'm really interested in understanding the science more and using these kind of the kind of detritus or artifacts of science that might be missed or seem mundane, to kind of reimagine them in a way that can engage people in science in different ways. And also to kind of twist these things that seem mundane or seem like detritus and reimagine them as something very beautiful, or that can actually, something that can evoke wonder to engage people with science in different ways.
And do you feel like the feces, for instance, can do that?
I do. I do. I mean, the piece, you know, it begins to shake in ways that is unexpected. So, and then people start to anticipate if a certain, if the shaker will start to shake based on people's Twitter activities. So it has different layers. You can sort of focus on the Twitter activity or the shaker itself or what's inside the shaker and where that material came from.
I mean, it doesn't remind me of this at all. It's totally different. But in the way that Chris or Phil uses the elephant dung as stands for the paintings, there's a sense in which everything is useful.
Yes.
Right.
Yes.
Everything, nothing should be thrown away. Everything should be utilized in some way.
Yeah. And I think that a lot of that sensibility comes from my relationship to craft too. And growing up in a family where everybody was a crafter and was using scrap material to make quilts and, you know, repurposing materials for textiles.
Yeah, and it is very craftsy, and the scientists, I don't know if you see, don't get to be so craftsy all the time.
Yeah.
I know we're on a very tight schedule, so I'm going to kind of rely on John to flag me when it's.
I might just jump in. So I think, so Laura, that was amazing. And you had me at llama. I was sold. I was like, yes, this is great. I'm really interested in, you don't see doilies so much, which is great. Like they're all over my mom's house. So like, why did you choose that? Like, can you tell us a little bit about, you mentioned your family are all crafters. So why choose that?
Yeah, I was really, working kind of simultaneously looking at a lot of microbial imagery. And as I mentioned, like SARS was really in the news and there was a lot of fear about it in the first outbreak in 2003. And I had also just been introduced to computerized embroidery, and I knew that I wanted to do something with it. And you can embroider with the machine in different ways, but I was particularly drawn to the idea of freestanding lace. And the more I researched the conventions of the radial doily and the traditional lace patterns, the more I realized that these enveloped viruses and these coronaviruses were not that far off from some of these conventions of doilies. So I just basically kind of figured out how I could impose the structure of these viruses onto the tradition of the doily.
Yeah, that's amazing. So thank you for sharing that. So just in the interest of time, we are